All right, so we have our last lecture here on glycogen metabolism. And this kind of doesn't have too much to do with glycogen specifically, but remember when we talk about the need to remedy low blood sugar levels, we've talked about this top-down and bottom-up approach. So the top-down approach really is taking starches, whether they be animal starch like glycogen that you have stored, or starches or sugars that you consume from your diet. Ultimately, that remedies low blood sugar, and then that glucose can then go through glycolysis to provide the energy that you need. So the premise here now is what if you have run out of your glycogen stores? What if you haven't eaten in a few hours? How do you remedy those low blood sugar levels? And this is that bottom-up approach. So this is called gluconeogenesis, and it's your body's way of converting things that aren't carbohydrates into glucose because you need to maintain that 5 millimolar blood sugar level. So the good news is that it's pretty much the reverse of glycolysis. So 10 steps of glycolysis that you learned, we're going to have 10 steps that we think about for gluconeogenesis with 3 new steps. Now here's where we have to remember that there was 3 steps that were far equilibrium in glycolysis, steps 1, 3, and 10. Anything that was far equilibrium, remember, had a large negative delta G. So a large negative delta G means it's a downhill reaction. We can't go downhill using the same pathway, and go in the opposite direction. So 3 of those reactions will have to use a different enzyme, and so it's a different pathway. So we'll have to kind of learn about the details of those 3, and really it's only the 10th step in glycolysis, the first step in gluconeogenesis, that we'll have to spend a little bit of time learning about. Okay, so we'll get into the details. 3 things that we'll talk about, kind of big picture details with our activity is, well, how do we reverse this 10th step of glycolysis, the first step of gluconeogenesis? We'll talk about the details of that process, and it has to do with making this molecule that we call oxaloacetate. The second kind of big thing that we'll talk about is regulating gluconeogenesis, which also means regulating glycolysis. And so we'll talk about an important branch point or metabolite. I shouldn't say it's a branch point metabolite. One that we haven't learned about yet is called F26P. So we'll learn about how that molecule regulates both flux in glycolysis through PFK and gluconeogenesis with FBPase. And so we'll learn about that in our activity. So those are the 2 main things that we'll learn about in our activity. So I'm going to go through our slides for tomorrow's lecture, kind of in an overview sense, so I'm going to skip over some details that we'll get to in tomorrow's class. Again, reversing this first step of gluconeogenesis means making this molecule that we call oxaloacetate. We'll see that molecule again when we talk about the citric acid cycle. But really, we can think about this as being an activated pyruvate. If you look at this molecule right here, it is a beta-keto acid. If we decarboxylate this guy, here's what it looks like. And this, we're going to have this guy having the nucleophile here. So if we decarboxylate here, we'll have this guy being nucleophilic, and then we can add whatever we want onto it. We'll end up adding phosphate, and it makes phosphoenolpyruvate. So again, we'll talk about those details in our activity tomorrow. So here again, we can see the 10 steps of glycolysis on the right. And again, steps 1, 3, and 10 being the large negative delta G steps in glycolysis. Those become the difficult reactions to reverse or I shouldn't say difficult reactions, the reactions that need to follow a different pathway when we reverse in gluconeogenesis. Okay, so we'll talk about the details of these 3 steps in our activity tomorrow, but big picture is that we can't use the same enzyme. You'll notice the rest of these 7 steps all use the same enzyme. Because it's not a far equilibrium reaction, it's easily reversible using the same enzyme. So only 3 steps being far equilibrium need to use a different enzyme. So this is the enzyme that is responsible for reversing that 10th step, which was again pyruvate kinase in glycolysis. It's actually a combination of 2 enzymes to get back to phosphoenolpyruvate. So this is going to become part of our activity that we'll talk about in class tomorrow. The details for how you add CO2 back to a molecule, and this is called carboxylation, so we have a carboxylase enzyme, and then how we do this phosphorylation chemistry, and this is an enzyme called PepsiK. So we'll talk about the details of those in our activity. So I'm going to gloss through these slides because we'll get to the details. Tomorrow we learn about another B vitamin that's important, vitamin B7, also called biotin. We'll see this anytime we have to do carboxylation reactions. So we'll talk about vitamin B7. Again, this is a way to utilize bicarbonate and make it into a CO2 that we could add on to another molecule. So part of our activity tomorrow is this slide. Again, having oxaloacetate is a way to think about having an activated pyruvate. So again, thinking about having pyruvate here, we're going to add CO2 to it and we get to oxaloacetate. Again, oxaloacetate, I said that's a beta-keto acid. So when we decarboxylate it, we get to an enol intermediate. Here we can see this with this mechanistically down here. We'll go through this in our activity tomorrow. When we decarboxylate this beta-keto acid, we get an intermediate which is capable of nucleophilically attacking the electrophilic phosphorus, that gamma phosphate of GTP, and then we get back to phosphoenolpyruvate. Again, details of that tomorrow in our activity. One other thing that we'll introduce, we won't spend a lot of time talking about this, is this idea that when we have things happening in the cytosol versus the mitochondria, do we actually move things back and forth? Or do we actually have separate enzymes? And if you remember, when we're thinking about what we do in lab, we're talking about the fact that there's cytosolic and mitochondrial versions of different enzymes. And so we have to think about whether that's the case or do we need to transport things across. And so we'll spend a little bit of time in our activity talking about shuttling systems that allow us to move things back and forth. Importantly, look here, we do not have transport mechanisms for oxaloacetate. What that means is we can disguise it through some chemistry, some biochemistry into different molecules. We can get those across. All right, this first step, reversing this first step, is what's problematic in gluconeogenesis. So we'll spend a lot of time in our activity working through that. The rest is actually fairly easy. So again, any step that we had a fire equilibrium reaction, we have to use a different enzyme, but for the rest of those 7 steps, it's the same enzymes, just reversing them. Big picture substrate cycles. Remember, we can talk about a substrate cycle in the context of a single enzyme pair. So again, PFK and FBPase. But we can also talk about substrate cycling in terms of an entire pathway. So glycolysis, gluconeogenesis. So this is not, as is any substrate cycle, without cost. So again, the cost of kind of going back through this process is a net of 4 ATP in terms of a cost. This I'm not going to talk about a lot of details. This will be something teased out in our activity. Big picture, F26P is an important metabolite that's responsible for regulating the activity of both PFK and FBPase. Again, these are the rate controlling enzymes of glycolysis and then gluconeogenesis. So levels of F26P control the activities of these enzymes, and the activities of these enzymes determine whether we are going through glycolysis or gluconeogenesis. So again, we'll break out those details in our activity tomorrow. Again, details for that on this slide I'm not going to cover right now. Just goes through how we synthesize and break and degrade F26P. And part of what we need to get to the point of doing is starting with a need state or a fed state signal and kind of talking about how it affects all of these pieces. So part of your activity and then part of exam questions that you'll see is walk me through the process of what happens when you have a low blood sugar signal and what that happens in terms of hormonal signals, intracellular signals, phosphorylation cascades, and in this situation, ultimately the concentration of this new important allosteric regulator. So we'll talk about the details of that. The last little piece to kind of talk about in this chapter has to do with the synthesis of other carbohydrates, right? Because glycogen is not the only carbohydrate that you have in your body or that other animals or other organisms have in their body. So the idea is, how do you create those glycosidic bonds if that is an endergonic process, a process that requires energy? And big theme is, well, you know what? We saw this with glycogen. You activate sugars by putting on an NDP, a nucleotide diphosphate. It's a good leaving group. So we'll talk about a few other examples of NDP sugar polymerization. We'll talk about it in context of mammalian systems as well as plant systems. So a couple of examples there. And the interesting mammalian story we'll talk about is lactose synthesis. And it has to do with hormonal signals. We'll talk about kind of an interesting story of how men have actually been able to lactate by the production of lactose, and again, a hormonal process. So again, that's all that we have for the rest of our chapter here on glycogen metabolism with gluconeogenesis, F26P, and how that regulates those processes, and then other carbohydrates.